Okay, and so now we're going to talk about transcription in eukaryotes, because we'll see that eukaryotic transcription, as can be imagined, because eukaryotes are a lot more complicated than prokaryotes, so too is this transcription, right? So the transcription in eukaryotes is a lot more complex than prokaryotic transcription. Though we do still see the same fundamentals that we find in prokaryotic transcription. I'm trying to find out which section it is. So we're on 12.10, and we're going to go 12.10 and 12.11. All right. So so transcription in eukaryotes differs from prokaryotic transcription. So it's going to differ from bacterial and archaeal transcription in several ways. First off, you have to remember just one of the main things that distinguishes eukaryotes from prokaryotes, in the sense that they have little organelles, right? They have compartmentalization. So in eukaryotes, we're going to find a nucleus, and inside that nucleus, this is where we normally find, we would find our nuclear DNA, right? So, and we know that the DNA is confined to the nucleus, but we know that we have this intermediate mRNA that's able to move from nucleus to the cytoplasm, because it's indeed in the cytoplasm where we have protein synthesis, where we have the translation machinery like the ribosomes. So we have that added step of actually physically moving our mRNA, our transcript, once we've undergone carried out transcription, we have that added step of moving it outside of the nucleus. Well, prokaryotes don't really have that, right? Prokaryotes, yes, have their DNA in this region. So it's a region, it's not a membrane-bound organelle, a region known as a nucleoid, but the DNA is found suspended within the cytoplasm. So there's no actual physical movement. They don't really have to go from one place to another. The DNA transcribes into mRNA and it goes into translation. In fact, a lot of times transcription and translation in prokaryotes are simultaneous. Like as soon as the mRNA is made, that first little bit of mRNA that is made gets immediately translated. So it's very fast. It's one of the reasons why bacteria are so quick to adapt to different kinds of environments as opposed to us. So it occurs within the nucleus, unlike prokaryotes. So the mRNA has to leave the nucleus for translation through the nuclear pores. In addition, our own DNA, since it's a lot bigger, it has, remember, we have that association with proteins. That association is known as chromatin, right? We have the DNA wrapped around nucleosomes and histones, wrapped around histones, and to form those bead-like nucleosomes. So the chromatin must uncoil to make the DNA accessible to the RNA polymerase. And other regulatory proteins that we need in transcription. Initiation and regulation require more extensive interactions between cis-acting DNA sequences and trans-acting protein factors. So we've mentioned a cis element, so an element that's found on the DNA in prokaryotes. We've talked about promoters, right? And the specific -35 and -10 sequences, or the -10 is also known as a Pribnow box. But eukaryotes have even more stuff. We have other things on the DNA that can either be found upstream of the gene or downstream of the gene. It could even be found a ways away from the gene, and it can still influence the gene's transcriptional regulation very much. And these are known either as enhancers or as silencers. So again, they are cis-acting elements because they're still DNA. They're still on that same DNA strand, right? Or on another chromosome even as well, but they're still on the same DNA, and they can influence a gene from being transcribed or translated, and then translated, obviously. And then we also have more trans-acting protein factors. So prokaryotes, this is something that your book doesn't really explain, but in prokaryotes, we do have trans-acting protein factors. Briefly, we mentioned the Rho termination factor, which helps with termination of the transcription. And so they do have some trans-acting protein factors, but with eukaryotic transcription, we have a lot more. So these trans-acting means that they're not DNA, they're made out of something else. That something else is proteins, and the proteins are going to are literally enzymatic, and they are able to directly associate and work with DNA. Right, so they're able to regulate and help the DNA being transcribed or other things. We're going to see lots of different kinds of transcription factors. So transcription factors, also known as transacting protein factors, TFs. Yeah, and then we have enhancers and silencers, which are called control units. So in addition to the promoters, we have these guys, these cis-acting elements on the DNA. In addition, then, when we first make our mRNA transcript in our nucleus, if we compare our mRNA transcript in our nucleus to the mRNA transcript that we actually find in the cytoplasm of that same transcript, the initial nuclear mRNA transcript is a lot bigger. And then the one that we find in the cytoplasm, the one that's going to go ahead and undergo translation, it's a lot shorter. So we actually take off a lot of what we call intervening sequences from our initial mRNA, and we'll go through that process in just a second. And we have to kind of tweak our primary mRNA transcript just a little bit. So as soon as we get our first mRNA transcript, so we have our DNA, it unravels and RNA polymerase synthesizes the RNA transcript, is known as a primary mRNA transcript. It's primary because we still have to work on it a little bit more before it can exit the nucleus and go into the cytoplasm. And so one of these things is to get rid of some of these intervening sequences and to further tweak the mRNA at its end, therefore to basically protect the mRNA, because we have a lot more enzymes that quickly degrade the mRNA in eukaryotes. So we do this by adding a little 5' cap. I literally think of like a little hat. Sorry, 5' cap, and then an additional 3' poly A tail, and then the excision of these, like excision or the removal or the splicing even of these introns or these intervening sequences. So we'll get into the details of every single thing, right? So eukaryotes possess a lot more forms of RNA polymerases than bacteria. So remember, bacteria, the way they can kind of decide which gene gets transcribed and which one doesn't is because of the different sigma factors that they have. And if you remember, sigma factors are simply a subunit of the holoenzyme RNA polymerase. And now with eukaryotes, we just simply have completely new RNA polymerases just from the get-go. So we have RNA polymerase 1, 2, which is the one that's most understood, and RNA polymerase 3. And they work on different things, right? So they work on different things. So usually RNA polymerase 1 is the one that usually makes, that transcribes genes that encode for rRNA, so the RNA that is necessary for ribosomes. RNA polymerase 2 is the most understood, and it's the one that usually encodes most, helps transcribe the majority of the genes that we have, the ones that become mRNA and then encode proteins. And then we have RNA polymerase 3, which makes some rRNA and which transcribes some RNA and actually tRNA. So RNA polymerase 2 is responsible for the transcription of a wide range of genes in eukaryotes, predominantly the genes that encode actual proteins. So you guys know that sometimes genes don't encode proteins. Sometimes genes stop at they just encoded rRNA or tRNA. That's when we have RNA pol 1 and RNA pol 3 working. RNA polymerase 2 is dependent on cis-acting elements and trans-acting elements. So both elements that are found within the DNA, that are part of the DNA, like the promoter, the enhancer, and the silencer, and then trans-acting transcription factors, so proteins that come and work alongside RNA polymerase 2. You'll actually come to see that RNA polymerases in eukaryotes are very dependent on other kinds of proteins in order to actually allow transcription to happen, while RNA polymerases in prokaryotes simply just attach. They don't really need too much happening. So the core promoter is just like in prokaryotic transcription, the core promoter is before the actual gene, the encoding gene. So upstream of the encoding gene, upstream of the transcription start site, we have a promoter. And again, this is where your polymerase is going to bind and where transcription is going to happen. So the promoters determine the initiation site and even the efficiency of the RNA, of the transcription. We do have a few kinds of specific elements and consensus sequences in eukaryotes. We have one that's very similar to the PRIBNOW element or sequence in bacteria. It's called the Tata box, or also known as the Goldberg-Hagenus box. But to be frank, honestly, not many people say Goldberg-Hagenus because that's a mouthful. Usually it's called the Tata box. And that's found at -30. So it's found 30 nucleotides prior to the transcription site. So it's like this transcription start site. It's a consensus sequence. The sequence and the function are analogous to those found in the PRIBNOW, the -10 the promoter region. Promote region. Let's fix that. Promoter. Okay, I spelled it wrong. There we go. I'm just gonna stop. Okay. We also have other cis-acting elements like enhancers. As their name suggests, enhancers enhance or increase the transcription level. And then we also have silencers. These decrease the transcription level. These could be very proximal to, like, so found near the promoter. Other times they can even be found very, very, like, far away from a distance, like quite far away from the gene that they actually regulate. So proximal promoter elements, enhancers and silencers. Enhancers and silencers can be quite close and proximal to the promoter, otherwise they're found really far away. And then in addition, we have other elements called proximal promoter elements, which are found right near the promoter. They often act in response to the cell's environment or at a particular time, right? So just depending on, like, even like the development. So at a critical stage of the development of an organism, they have to express certain genes. Those genes are going to be regulated with the use of enhancers and silencers. So their transcription and therefore their expression is going to be activated or deactivated depending on the enhancers and the silencers. Each eukaryotic gene has its own unique arrangement of proximal promoter elements, enhancers, and silencers. And actually, I believe we're going to get into this in chapter 50 when we talk about regulation, which is a really crazy chapter. But sometimes enhancers and silencers can be shared with genes depending on, like, if the genes carry out similar functions. So yeah, so a lot more stuff going on compared to at least compared to prokaryotic cells, prokaryotic transcription. We also have transacting elements. So these are basically your transcription factors that help and aid the RNA polymerase to actually work. So for example, RNA polymerase 2 isn't actually going to even be able to bind to anything without the presence of transcription factor 2D, which is, I don't know if I mentioned it. I don't know, anyways, it's there. So without a particular transcription factor, it won't be able to actually bind to the promoter in the first place. So transcription factors usually fall into two broad categories. We have the general ones, right? So, okay, here, yeah, general ones. These are absolutely necessary for RNA polymerases in order to actually fulfill their transcriptional role. So without them, RNA polymerases won't bind. So it just won't bind. In order for the RNA polymerase 2 to bind directly, to bind to the promoter, it needs to first have a specific transcription factor 2D. There it is, yes, there we go. Transcription factor 2D, which has a TATA binding protein, right? It has a TATA binding domain on it. So a part of it's going to bind to the TATA box, that -30 sequence, right? And then once that specific transcription factor is recruited, is bound to the DNA, it actually recruits other transcription factors like transcription factor A and B, and then, so followed by other general transcription factors. And then, and only then, does RNA polymerase 2 actually bind. And so once we have all of these proteins and enzymes working together and bound to the DNA, at that point we have the pre-initiation complex, which is basically what determines the transcription start site. And as soon as that's made, we can actually go ahead and start elongation or initiation and elongation of the transcription. So then we also have specific transcriptional activators and repressors. These ones they don't actually, their presence doesn't inhibit transcription and it doesn't activate transcription. So they're not like on and off switch like the general transcription factor. So their presence allows transcription to happen. Their absence inhibits transcription to happen, because without them, RNA polymerase can't bind to the DNA, bind to the promoter, and therefore transcription doesn't occur. Specific transcriptional activators and repressors, on the other hand, are more like light dimmers. So they basically influence the efficiency or the rate of the transcription. So they make the transcription faster, or they minimize and decrease the transcription so the gene doesn't get transcribed quite as much. So it's still a way of regulating it, just not as, it's not like a light switch, it's more like a light dimmer. That's a pretty good way of saying it. So they bind to, so in order for them to not directly inhibit or activate transcription, they just affect the rate pretty much of transcription. They do so by binding to the enhancers and the silencers. Remember those cis-acting elements. And that way they can regulate transcription initiation by aiding or preventing or making it harder, basically, for the pre-initiation complex to assemble. Okay, So heterogeneous nuclear RNA and its processing. So when mRNA is first made, remember it's called the primary transcript, and then that primary transcript has to be tweaked a little bit, actually a bit, before it can exit the nucleus and then be actually read by the translation machinery. So heterogeneous nuclear RNA is RNA molecules found in the nucleus, including pre-mRNAs or your primary mRNAs, right? So they have to be modified a little bit in order for the mRNA to be completely usable, and we call those the usable translatable mRNAs, mature mRNAs. So in eukaryotes, there are post-transcriptional processes, so post-transcriptional modifications that have to happen to this pre-mRNA before, or primary RNA, before it can leave the nucleus. So it's different in between textbooks and whatever. You can find primary mRNA or pre-mRNA, I believe. Since it's in my slides here, your textbook uses pre-mRNA, so that's what I'm going to try and use from here on out. Okay. So the first thing that actually happens, I'm going to change the video. One of the first things that happens is at the 5' end of the mRNA to prevent the possibility of it being degraded by exonucleases, right? Because we do have exonuclease activity that can attack the mRNA and get rid of it. So if I have my DNA right here, right? And then this section right here, I have the template DNA gets read, and I make my complementary mRNA strand, and then it comes off here. Then here I have my mRNA, right? But this is still in the nucleus. Let's make the whole cell still in the nucleus. So this is my pre-mRNA. Now, inside the nucleus itself, we do have enzymes that can attack and degrade the mRNA. So they chomp at the sides, at the ends of the DNA. They are not endonucleases. Endonucleases are able to actually chomp inside, like in the middle bases, so not at the linear ends, the bases in the middle. We don't have endonucleases. We do, however, have exonucleases, which basically eat and chomp at the mRNA on the extremities or on the ends, so exonucleases. We do have exonucleases in the nucleus, and so usually this would mean that my mRNA would basically be eaten at both ends. However, that doesn't happen because our pre-mRNA is going to have some post-transcriptional modifications. The first one is a 5' cap. So if this is my 5' end, right? This is my 5' end. I'm going to have a little 5' cap, which is essentially just a methylated guanosine, right? It's just a guanine nucleotide that has been methylated. And this acts as a little cap that just kind of protects this end right here from any kind of degrading enzymes. And then on the opposite end, I have my 3' end. Here, in order to help protect the mRNA from rapidly degrading, we just add a poly A tail. Now this tail it could be a huge, so it's just adenines repeated over and over and over again. It could be incredibly long. It could be up to like 200 to 300 adenines, and that's why we call it a poly A tail. And so both of these structures help they help basically preserve and lengthen the lifetime of the pre-mRNA, right? And so we're kind of doing these post-transcriptional modifications on it. In addition, they also help allow the pre-mRNA to exit through the nuclear pores. There's not a lot of concise information on this, but it said that they're assuming, or they're still looking for evidence that the 5' cap has to associate with some of the nuclear pore proteins that we have going on here that make the nuclear pore, right? Both the poly A tail and the 5' cap, they associate with the nuclear pores proteins to allow the mRNA to exit outside. Okay, and then the last and final thing is that within the mRNA itself, the pre-mRNA itself, there's a lot of intervening sequences. Basically, these intervening sequences are useless. We don't need them. They're not actually essential to make the protein. In fact, if they are read and made into a protein, our protein will be non-functional. So we have to get rid of them. We have to get rid of them. And then the essential, that's a weird thing, sorry. The essential, here we go. The essential parts, okay, called the exons, have to stay in, and they have to kind of be spliced together, right? So this would be exon 1, exon 2, and exon 3. We want to keep the exons, remove the introns, okay, and then splice the exons basically together, ligate them, put them together. And so at the end, our mature mRNA is going to be a lot shorter than our pre-mRNA, and with all of these kind of modifications done to it. So if this is my mRNA, you can see it's a lot shorter. Should have made it. That's actually not a very good representation, but let's pretend this is really long. Okay, and the 5' cap is right here. Okay, so it's a lot shorter than your nuclear pre-mRNA because we only have exons here. We only have the essential good, like the actual part that encodes the protein. And then we have our 5' cap, our methylated guanosine, and then our poly A tail, which also continues to protect the mature mRNA from any kind of exonuclease activity. Once we have our mature mRNA, this is the only kind of mRNA that can actually be read and translated by our ribosome and our translation machinery. Okay, so this is what it looks like. And so a lot of students get introns and exons confused. The way I remember is that exons start with an E, so they are essential. It's what I've been trying to say this whole time. So they're the essential part of the gene that gets actually translated into protein, and then introns are intervening. A lot of students think that introns stay in the mRNA. That's incorrect. Introns are removed from the mRNA. Okay, they're removed from the pre-mRNA, and that's called, that whole process is known as splicing. We'll get into splicing in just a second. So if you see here, right, introns are intervening sequences, are regions of the initial RNA transcript not expressed in the amino acid sequence of the protein. The DNA sequences are not represented in the final. So basically, these are DNA sequences because they're still encoded by the DNA, but they're not represented in the final mRNA product. So you may be thinking, what the heck is their point? We still don't have an exact idea as to why we have so many introns. There's some genes out there like collagen. About 85% of the gene that encodes collagen is introns, is intervening sequences, which is like crazy. So it only has like 15% is exons or relative essential code. So what's the point? There's a lot of indication that they're very useful for regulation of like transcription. So like turning it on or off, right? And I think we might go over that in chapter 15. We'll get there. Probably. So genes that contain them are called split genes. Basically, every single gene in eukaryote or like in humans are split genes. There's only a couple of genes that don't have any introns. Those and it's kind of strange. Those include things like interferons, right? Interferon genes, or even interferon genes, or even, oh my goodness, I forgot the other gene. What's the other kind of gene? Histones. Histones, sorry. Histones don't have any introns. Okay, so exons are sequences that are retained and expressed. They're expressed exons, essential, okay? Prokaryotes do not have introns, so that's a little bit of a mistake. They don't have introns in the sense that most of their genes don't have these intervening sequences, but some of their genes that encode for tRNAs do have like some of their sequences are removed before the tRNA can be functional. And so though we don't like to call them introns, they do have some, like the genes that encode the tRNAs have some of these intervening sequences. But for the most part, prokaryotes don't have them, right? But it's interesting to know that even like viruses that infect animals or eukaryotes do hold introns, and so they still rely on our own like machinery, specifically the spliceosome, which is what we use to remove these introns. So evidence of the electron micrographs of heteroduplexes. If you remember, heteroduplexes are just when we have DNA that is still being transcribed, and so it has a partial RNA transcript attached to it. And you can basically take the DNA that we transcribed, that length of DNA, and try to hybridize it with the mature mRNA that we then isolate from the cytoplasm. And what you'll notice is that the mRNA, because we've removed all of those intervening sequences, is a lot shorter than the initial DNA where it came from. And so they are complementary to each other. We form these heteroduplexes, but those places where we remove those introns, and the DNA obviously still has them, right? The DNA loops out. And so we get all this weird loopy kind of heteroduplex that kind of issues. So this is kind of an evidence from the electron micrograph. So we have post-transcriptional modification. Introns are removed by splicing. Exons are then joined together in the mature mRNA, and then the mature mRNA is smaller than the initial mRNA. The precision is like of this process is incredible, because if you do a mistake while cutting or splicing and then putting the exons together, right, that can be highly, like, it can cause a lot of problems. It can be a humongous like mutation, and it's a place where a lot of errors could be introduced. Amazingly enough, though, it's extremely precise, and not a lot of, actually, yeah, a lot of errors are not introduced. Most eukaryotic, oh, there we go. Most eukaryotic genes have introns, only a few of genes lack them. Exceptions include histones and interferons. So if only I had read here, that would have made it a lot better, and I wouldn't have had to rely on my terrible memory. Okay. Right. So this is just an example, right? This is like the gene that encodes mouse insulin. You can see there's 2 exons here and then 1 intron. So like an intron of, like, it's significant, right? So we have to remove that in order for the insulin to actually be able to be to make mature mRNA, and therefore translatable mRNA. The beta globin was one of the first genes that we actually realized this in, like we saw. And then the albumin, okay, of the egg, of the chicken egg, you can see all of these introns. Yeah, like I said, collagen has like one of the most, like, is one of the most intron saturated gene there is, and it has about 50 introns. So it doesn't really matter how many there are. That doesn't change anything specifically about the gene. It just, there's a little bit more work happening in order to make our mature mRNA. So the splicing mechanism. So there's a couple of different kinds of splicing mechanism, and it first off kind of depends on where the mRNA is. So for a lot of like, if we have the bacteria, right, so the prokaryotes, where we know we don't really have introns, but sometimes some sequences are removed specifically to make your tRNA. And those kind, like in that situation, or in lower levels of eukaryotes, and even some plants, which are kind of higher levels, I know, but a lot of lower level eukaryotes and some plants, we can have self-splicing, like different kinds, like self-excising RNA. So basically, the introns can get rid of themselves without the use of any kind of proteins or like outside transacting elements to help them out. These are known as group 1 and group 2 introns. So self-excising. So again, introns are categorized into different groups. Group 1 and group 2 are able to self-excise, and then group 3, which are the ones that usually have, like, that most eukaryotes have, the ones that you have. So where you have to take your nuclear mRNA and get rid of all of its introns. Usually it's mediated by a far bigger complex, by a far bigger, like an actual enzymatic protein/RNA complex called the spliceosome. So group 1 introns and group 2 introns can self-excise. So the intron, literally, they self-excise. So they kind of get rid of themselves all by themselves. This is possible because, if you remember, RNA actually has it does have a catalytic role. It can carry out like catalysis. This is known, like, these are called, when the RNA is involved and has catalytic activity, they're known to be ribozymes, right? And this can be seen with like introns splicing themselves out. And we're actually going to see that in the spliceosome, that big complex, which is made out of protein, that takes the mRNA introns out. It's not solely made out of proteins. Actually, one of its main components is the fact that they have these small nuclear RNAs, and they help basically allow for the function of the spliceosome. So they have this ribozyme component as well. Yeah. So, okay. So the simplest mechanism for intron removal is at the endonucleases. Remember, endonucleases, they cut in the middle of the nucleic acid. They cut each end of the intron. The introns are removed, and then the terminal ends of the adjacent exons are ligated, are put together through RNA ligase. Very similar to DNA ligase, except it's RNA now, so it's called RNA ligase. It occurs in tRNAs and bacteria. Mechanism is more complex in higher eukaryotes. So here we have group 1 introns and group 2 introns, which basically carry out this very simple mechanism. It's a self-excising mechanism. There's no additional components necessary. It occurs in many lower level eukaryotes like protozoa. And then you basically, yeah, oh, here we go, bacteria, lower eukaryotes, and higher plants. Well, okay. And then they just kind of take themselves out, right? They have specific nucleotides in, like, especially like guanine is one of the, like, a very important nucleotide that's able to actually carry out transesterification reactions. You don't have to know the specifics of it, but they just, do you? Not really, no. They just kind of take themselves out, no problem. And this is just a very simple kind of mechanism because you don't have any other additional components. Then we have the other ones, the group 3s, the nuclear mRNA introns, which are much longer, more plentiful compared to group 1 and 2, and they're found in the eukaryotes and stuff. And then these ones are mediated, the actual intron removal is mediated by the spliceosome. So the spliceosome, let's look at it. So it's made out of proteins, and it also has a unique type of RNA called small nuclear RNA, right? Which is complex with proteins to form SNRPs. This is literally pronounced SNRPs. SNRPs are really important because the small nuclear RNA is very uridine or uracil rich. And so sometimes we just call them all the different SNRPs that we have in the spliceosome. We just call them U1, U2, U3, U2, U2. U1, U2, U3, U4, U5, U6, etc. Right? And so these uridine-rich ones are like uridine-rich small nuclear RNAs actually are able to bind to specific sites on the intron. So introns have a very specific sequence to them. In fact, one of the most tedious things that you may, I don't know if you do any kind of programming, or not programming, but just DNA sequencing, and if you go into the lab, is finding introns. Well, finding introns, and from your DNA sequence, which is going to be transcribed into your mRNA if you're working with eukaryotes, okay? Finding the introns, removing them, and then getting your actual DNA sequence that completely codes for your protein. And you can do that because, again, you have specific sequences and like arrangements of these sequences on the intron. And so at the 5' end of the intron, you'll find a GU donor sequence. You'll find a branch point adenine in the middle area of the intron, and then at the end, at the 3' end, you'll have an acceptor sequence known as the, which is basically carries an adenine and a guanine. Okay, so these are consensus sequences, right? They're found in every single intron. And these are the sequences that your small nuclear RNAs and the SNRPs, your U1, U2, etc. These are the sequences that the SNRPs in the spliceosome are actually able to attach to, right? So U1, for example, is complementary to the GU donor sequence on the 5' end. Then once the U1 binds, the rest of the spliceosome follows suit. Don't need to know the order of the spliceosome forming, right? Because remember, it's this huge complex. And then the actual splicing occurs. At this point, actually, you do need to know kind of how the splicing happens. Let's move this away. The branch point, the A in the middle of the intron, attacks the GU, the 5' splice site at the beginning of our intron. And this starts forming kind of like this weird hook lariat. Okay, this lariat form, and then at the end, once this lariat is formed, this first part can then, the 5' splice site can then go ahead and attack the 3' splice site, and so then we excise our full intron completely out, and our intron is excised in lariat format. So exons are then ligated. Once we have our exons are then put together, right, without the intron intervening and basically interrupting everything. And then the nuclear processing represents a potential regulatory step in gene expression in eukaryotes. That's actually a huge, not potential, it's a huge regulatory steps in eukaryotes. We can even get, I don't know if I have to talk about this, but different kinds of genes can be, not genes, but different kinds of mRNAs can be made from one gene, which gives us the potential of making more like different kinds of proteins, because sometimes our exons aren't put in order, like aren't ligated in order. If I have exon 1, 2, and 3, you would think that exon 1 and 2 will bind, and then exon 2 and 3 are going to bind. That's not necessarily the case. Sometimes things can mix up. So exon 1, which normally is here, 2 and 3, 1 can jump over here. Now I have 2, 3, and 1 that get ligated. And just the mixing up of these domains, right? The exons that will then be translated into protein domains can give us new proteins. And so this is a really critical regulatory step in gene expression. Oh, there we go, alternatives. And this is known as alternative splicing. So alternative splicing produces related proteins from single genes, increasing the number of products derived from a single genome. So you can see here we have how many exons? 5 exons, and everything that's not highlighted or colored in, those are your introns. Your introns are going to be excised by the wonderful spliceosome machinery, and then these exons can be put together. And something I didn't mention actually is that sometimes not all exons have to be put together. Sometimes you can even like skip an exon. So here in this example, the first one, every single exon is accounted for. And then on the second example, we actually don't have exon 3. Here we don't have exon 4. What they don't show you here is that sometimes the order is also different. And this just, again, creates different kinds of proteins. These proteins, this is different kind of proteins. That's why we have, like humans have about 22,000 genes, and yet we have hundreds of thousands of proteins because we have this alternative splicing capability. I believe it was a gene in Drosophila. It was just one gene, but that one gene, just thanks to alternative, Drosophila is also eukaryotes, right? They're flies. This one gene, thanks to alternative splicing, could actually result in like more than 150 proteins. Crazy. I should remember what gene it was. Probably like some I gene or something like that. Okay. So, yeah. So I'll pick up with the last part in just a second.